Welcome back, students. Today we are going to discuss part 7 of Unit 9, that is application of biology. Before I start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. In today's video lecture, we are mainly focusing on immunity, innate immunity, adaptive immunity, active and passive immunity. Okay, immune response, what is antibody, what is vaccine and antisera, and genetically engineered insulin. So these are the topics which we are going to discuss in today's video lecture. So first of all, this chapter, what is the name of the chapter? Application of biology. Okay, so in application of biology, first of all, we are going to discuss about the application of biology, or we can say biotechnology, in medical field or in medicine, right? So biotechnology means bio plus technology. Techniques of biology used to create some kind of molecule, some kind of product which is useful for human. Okay, so in this application of biology, the recombinant DNA technology process, mainly this RDT, or we can say recombinant DNA technology process, has made immense impact in this particular area, healthcare industry. How? By enable mass production of safe and more effective therapeutics drug. With the help of recombinant DNA technology, in today's scenario, scientists are able to produce more accurate, more effective therapeutics drugs, which are of course safe as well for the human. Okay, so this recombinant therapeutics do not induce any unwanted immunological response. Although they are isolated from various other sources, means bacteria, yeast, or other kind of source, but still because of the technology, scientists nowadays are able to conclude that this particular drug is safe or not. Okay, so in this, with the help of this recombinant therapeutics, these type of drugs do not induce any kind of unwanted immunological response. At present in the market, about 30 recombinant therapeutics are available for the human use worldwide. And out of these 30 recombinant therapeutics, 12 of these are presently used in India as well. Okay. So out of these therapeutic drugs and product, one of them is this insulin, genetically modified or genetically engineered insulin. As you know, that diabetes nowadays, it is a lifestyle disease, diabetes and high blood pressure. So management of diabetes or management of adult onset diabetes is possible by only taking insulin, right, at a regular time interval. This is the only solution. So just think, if that much amount of insulin is not available in the market, which is required by a given population, then what will happen? That is a very panic situation, right? So in that particular situation, can you just imagine, is it possible to develop or isolate insulin from the bacteria? In that scenario, it is very easy to grow certain kind of bacteria, because bacteria are able to grow within 24 hours in an ample amount, right, under laboratory conditions, and where in that situation we are able to isolate insulin comparatively at a far speed, right? So management of diabetes onset, diabetes in adult is only possible by insulin, as I told you just now, okay, at a regular time interval. But if we are able to produce insulin in the bacteria, suddenly the whole process become very simple. You can easily grow a large quantity of bacteria under laboratory condition and able to isolate insulin, whatever amount we needed, right? So initially, with the help of this technology only, biotechnology, right? Initially, in initial days, insulin isolated from the pancreas of slaughtered cattle and pigs. But as you know, this insulin isolated from other sources other than human, means cattle. So some patients show some kind of allergic reaction for the same, because their body found it as a foreign particle. So non-self reaction, because of the non-self reaction, some allergic reaction was there in their body, right? So after continuous, because of the continuous research and different suggestions and different research and other aspects which was initially discussed by the different scientific community, nowadays after several years some companies were able to isolate insulin in bacteria as well. Okay, so but do you know that in mammals, including human, this insulin is also synthesized as a prohormone, just like enzymes in human. Okay, and this insulin consists of two short polypeptide chain A and chain B. Okay, and these two chains are linked together through a disulfide bridge. Okay, so like a proenzyme, as I told you, this prohormone also need to be processed before it become a fully mature and functional hormone. As you can see in this particular picture, this green one is the A peptide and this blue one is B peptide. And both of these peptide is linked together by disulfide bond, but between them there is another peptide molecule known as C peptide. Okay, so during maturation process, just removal of this free C peptide bond, C peptide has occur. Okay, so like a proenzyme, this already I told you, this prohormone, that is we can use the term proinsulin for the same, also need to be processed before fully functional form or fully mature form. So removal of this C peptide is must, which is present there as an extra stretch, okay, during maturation into the insulin. Okay, so main challenge, just now I told you that after some time, initially slaughtered cattle's pancreas is used for the isolation of insulin, but further some patients show allergic reactions. Research was going on continuously, but in front of the scientific community the main challenge was this, that this insulin contain two type of chain, B chain and A chain. How they can assemble these two chains into a mature form, this was the main challenge. Okay, but because of the continuous research and other activities within scientific community, one American company known as Eli Lilly in 1983 was able to produce this genetically modified insulin. Okay, what they did, they actually grow both the chain, A chain and B chain, in separate plasmids. They just ligate the component into the plasmid of E. coli. Okay, and as bacteria and transform it into the bacteria. As bacteria multiply continuously, plasmid will also get multiply. Basic principle, right? So this is how they were produced, chain A and chain B, in different portion of bacteria in separately. Okay, separately, chain A and chain B were produced separately, and then both the chain extracted by them and combined. After the extraction, both the chains were combined by creating disulfide bond to form human insulin. So this was the first company in 1983 who is actually synthesized genetically modified or genetically engineered insulin. Okay. Now let's discuss another important term which we must discuss in this particular video lecture, that is immunity. Okay. So as you know, every day our body are exposed to large number of infectious agents. These infectious agents or pathogens are present in air, soil, water, everywhere, right? But only a few of these exposures result in disease. Why? Only few. Because our body is able to defend itself, okay, from most of these foreign agents. And this defense capacity of our body is known as immunity. Okay, our body's host, or we can say ability of the host to fight against disease or disease causing organism is called immunity. Okay, we have this power inside our body. Okay, and further we can classify, to understand this particular term, we have to classify this immunity into two different categories. First is innate immunity and second is acquired immunity. Okay, so let's discuss innate immunity first. So what is innate immunity? Innate immunity is a non-specific type of defense. Non-specific type of defense. Okay, that is present at the time of birth. This defense system or this immunity or this power is present at the time of birth with different type of barrier. We have different type of barrier within our body. Okay, we can classify these barrier into four different categories. First category is physical category, then physiological category, then cellular category, then cytokine category. Okay, so first is physical category. What is this physical category or physical barrier? First line of defense of our body is our skin. This is first line of defense. This is the main barrier which actually prevent entry of the microorganism inside our body. Okay, if there is a cut in our body anywhere, okay, and we don't apply any kind of antiseptic or respective cream, skin cream, soframycin and all, then soon after that it will show some kind of swelling, pussing and other things. Okay, so if there is no cut, nothing, no microorganism is able to cross our this outer covering, or we can say skin. So we can say this skin act as a main barrier. Okay, some openings are there in our body, like nostrils, okay, like our mouth cavity. Okay, so and these cavities, these opening contains some mucus. These opening contains some mucus coating epithelial lining, and some tiny hairs is also present in our ear and nose, right? And similarly, these mucus coating is also available in our respiratory system, or as I told you, in nostrils, in our gastrointestinal system, and our urogenital system as well, which help in trapping microbes entering in our body, which very easily trap dust particle and microorganisms inside the mucus and the hairs present there. Okay, so these things, skin and mucus coating, act as a physical barrier for us. First type of innate immunity. Second type is physiological barrier. Second type of innate immunity is our physiological barrier. In physiological barrier, some kind of physiological parameters helps us to provide immunity, like presence of acid HCl in our stomach, presence of saliva in our mouth, presence of tears in our eyes. These all secretions actually prevent bacterial growth. Okay, so these secretions like acid, saliva, tears act as a physiological barrier. Clear? Third is cellular barrier. Cellular barrier means certain kind of cells act as a barrier, like leukocytes, or we can say WBC, white blood cells. These white blood cells like polymorphonuclear leukocyte, some kind of monocytes, some kind of natural killer cells, means different kind of cells. These are found in our blood. Okay, some like macrophages, these are found in our tissue, can phagocyte that microbial growth or pathogen and destroy microbes. So they also provide us a kind of immunity. They directly fight with the microorganism, right? And the last one is cytokine barrier. Cytokine barrier means these cytokines are certain kind of proteins, certain kind of protein molecules. Okay, these cytokines were actually present in these cytokines is present in the virus infected cells. Actually, virus infected cells secrete these proteins as a interferon, which protect non-infected cells. They just give a signal through interferon to the non-infected cell for further viral infection, and give non-infected cell as an alarm for further viral infection. So this is the fourth type of innate immunity. Four type immunity we have classified into two categories. Okay, first is physical, innate immunity type physical, outer covering, mucus. Physiological, different kind of secretions like acid, saliva, tear. Third is cell mediated or cellular, different kind of cells like WBCs, macrophages, phagocytes, these kind of cells. And then fourth one is cytokine barrier. Here some interferons, some protein molecules, which is secreted by the virus infected cells and protected non-infected cells. Okay, so these are the four type of innate immunity. Okay, now in this picture you can clearly see this immune system two type, innate one, and second type is adaptive one. We have discussed these three: physical, physiological, stomach, lysozyme, etc. Innate immunity, different kind of cells, and cytokines, right? Second type is adaptive immunity. In adaptive immunity, this adaptive response is mediated by different kind of cells of our body. Okay, different kind of cells of our body. So we can further classify adaptive immunity into two different types. First is cell-mediated response. Second is humoral response. Okay, humoral response is given by B cells. Okay, as these are present in the blood as such. So these B lymphocytes actually mainly responsible for humoral response, and these B lymphocytes further is able to produce antibodies which directly fight with the antigen or pathogen or foreign particle. In cell mediated immune response, some kind of cells, okay, T cells, some kind of cells which is produced by T lymphocytes get participate in the fighting with the pathogen, and this T lymphocyte will able to produce different kind of further cells which directly fight with the antigen. That's why we can use the term cell mediated response. Okay, here antibody, what is the last product? Antibody. Here what is the last product? A different kind of T cells. So this is, these two, cell mediated response and humoral response, are the type of adaptive immunity. Okay, once if we are infected with some kind of pathogen, after that our body is able to fight. Initially we get suffered with that particular type of disease. Slowly our body, our immune system recognize the type of pathogen, and quickly after some time, not quickly, after some time, our body is able to produce antibodies and our defense system against that. Okay, so in acquired immunities, means acquire after pathogen infection. Okay, that's why we can use the term acquired immunity is a pathogen specific, because it is characterized by the memory. Okay, what is the meaning of memory? It means that our body, when it encounter with a pathogen for the first time, so first time our response is of low intensity. We'd get suffer with that kind of disease, virus, cold, fever or whatever. Okay, with so our defense type is low. It is of low intensity, because before that we never have that kind of disease, like just COVID-19. Initially there is no vaccine, nothing, and that this pathogen is suddenly grab all over the community throughout the world. So initial response was very slow, very low intensity, so that is known as primary response. But once we were affected with the COVID-19, if same pathogen, same virus again come, and during second wave, that time most of the people show immunity for the same virus. Why? Because during the time span between primary and secondary, first wave and second wave, our body recognize the pathogen and prepare itself for the defense, for the fight, by producing antibody against COVID-19. Right? So acquired immunity is a pathogen specific. First of all, we have to understand this. Okay, and it is characterized by the memory. This means that our body, when it encounter a pathogen for the first time, our body produce a very low defense towards that pathogen, and that response is known as primary response. Okay, but subsequently, if our body encounter with the same pathogen again after some time, that time our body response is highly intensified, and that response is known as secondary response. Okay, and what this secondary response show? This secondary response show that our body appear to have a memory of the first encounter. Our body memorize that particular pathogen and produce accordingly the immune power or antibody for that particular pathogen. Clear? So the primary and secondary response, these primary and secondary response are carried out by different type of cells. Okay. And for that the two type of cells are mainly responsible for these kind of primary and secondary immune response. What kind of cells? B lymphocytes and T lymphocytes. Okay, this B lymphocytes is already we have discussed previously in that picture, is mainly responsible to produce antibodies in response to a specific pathogen, while T lymphocytes or T cells themselves do not create antibody but help B cell to produce them, and these are again produce some kind of cells, cell CD8, CD4, T helper cell, other kind of cells. That's why we can use the term cell mediated immune response by T cells, okay, and humoral immune response by B cells, right? Okay, now we can again classify immunity in two different ways: active and passive immunity. Acquired immunity further we can classify into two different ways: active and passive. When a host is exposed to antigen directly, naturally, we can say naturally, okay, which maybe this host in the form of living form or in dead form or any protein, okay, that time after exposure our body actively produce antibody against that particular antigen, okay, and this type of immunity is called active immunity. I'll repeat again: when a host is exposed to antigen which may be in the form of living or dead microbe or other protein, antibodies are produced in the host body, and this type of immunity is called active immunity. Okay, active immunity is slow and takes time to give its full effective response. It takes time to give its full effective response. Okay, while injecting microbes deliberately during immunization or infectious organism gaining access to our body during natural infection induce active immunity. Okay, so during immunization also, if we will incorporate weak antigen or any part of that antigen inside our body, that is again a type of active immunity. Okay, now what is passive immunity? Passive immunity means when ready-made antibody are directly incorporated in our body against that particular antigen or foreign agent. Means incorporation of antibody is known as passive immunity, in which our immune system is not able to engaged to synthesize antibody against that particular antigen directly. If we will incorporate antibody against that particular antibody, this is known as passive immunity. Passive immunity one can get from mother's milk during the time of mainly newly born baby achieve this kind of passive immunity from their mother's milk, because this milk contain yellowish fluid colostrum, and this during initial days of lactation, this particular yellowish fluid contain a particular type of antibody known as immunoglobulin A. So directly incorporation or directly transfer of antibody from mother's milk to baby occur during this process. So it is a type of passive immunity. Okay. Similarly, during gestation period, this fetus also receive some antibodies from mother. A kind of antibody, mainly immunoglobulin A, is able to cross the placenta during pregnancy. So this is how again that fetus achieve some kind of immunity by direct transfer of antibody from mother's blood to placenta. This is again second example of passive immunity. Okay. So acquired immunity we have classified into two categories. First is active immunity, in which our body is totally involved for the production of antibody. Okay. And passive, where our body is not involved, direct incorporation of antibody occur. We can further classify active immunity and passive immunity, two different types, natural, artificial. Here also natural and artificial. Naturally, active immunity we can involve in natural active immunity, in which natural infection occur. So naturally antibody is developed by our body in response to a infection. Artificial, when artificially we injected some kind of pathogen, weak or dead pathogen or antigen we can say, inside a body. So against them, our body quickly synthesize antibody. Okay, so this we can achieve through vaccination. From after the new baby birth, some kind of vaccination is a must, like BCG, DPT, hepatitis B. Okay, to immunize that newly born baby for various these deadly diseases, right? So artificially, in which artificially doctors injected weak or dead microorganisms inside a body. Okay, quickly our baby's immune system recognize those microbes and their body quickly synthesize antibody against them. Okay, so this is two type. Again, passive immunity, again we can classify two type, natural and artificial. Naturally, antibody received by the baby through milk, through placenta. Okay, when mother, just now we have discussed this. Artificial mean when after when antibody received by the patient as a injection, like antitoxins. Okay, so like gamma globulin, directly when incorporation of antibody occur. Okay, during a snake bite, during some kind of thing where we cannot wait for, naturally body can synthesize the antibody. We have to give direct antibody in that case inside the patient's body. So this is all about the today's video lecture. If you are having any doubt, any confusion related to today's video lecture, you can write down to me in the comment box. We will discuss later on. Till then, take care. Bye bye. Namaskar and keep learning.